So everything I show you, everything I give you, everything. So we're going to look at 2 animations today. They're both already on the Moodle. So if you want to go check those out later and see what's going on, you'll be able to do that. The live stream is working. All right, let's pray and we'll get started. Heavenly Father, thank you for this day. Pray that you give us minds that are ready to receive, to learn, to pay good attention, Lord, to focus, to honor you through our time here, and not to squander this opportunity. Father, help me to be just clear as I ought to be, and Lord, just to be gentle and kind and loving in all these ways. Father, be with us, we pray in Jesus' name. Amen. Well, today we're starting, so this is second semester, this is organismal biology. We'll be interested in some of the cellular stuff that comes in. It's going to come in today. There's just no way to get around it. We're going to look at these little tiny things that infect your cells. They don't have cells, but they can't live without your cells. Actually, they're not alive at all, frankly. They can't exist without your cells. And so some of the stuff that happens at the cellular level is going to be inevitable. And we're going to look at how your body handles them. And so that's going to be, again, coming back and looking at things at a cellular level. But by and large, everything we're going to do this semester is going to be a lot bigger than that. It's a study of organismal biology, definitely things that are alive, but we're going to start with something that's technically not alive, but has a huge impact on things that are, okay? And so we'll be looking at viruses, viroids, and prions. If you're looking for a title, that's what it is. Later on, if you were to look at your Moodle, that's the title of this lecture. Okay, so viruses, viroids, and prions. Everybody's heard of a virus. I'm guessing no one's heard of a viroid or a prion. Is that pretty accurate? Yeah, these are weird things. Viruses, they infect cells. It's what they do. It's how they exist. It's how they continue to exist. But they're not alive. We talked about these different characteristics of life the other day, right? Can you all help me out? We had 5 of them.
Oh, grow?
Yeah, they grow. Okay. What else do they do?
Uses energy.
You use energy. They move. What else?
Reproduce.
Reproduce. What else?
Respond to stimulus.
Respond to stimulus. Good. So viruses can do some of this, but they don't do all of this. They don't get bigger. Okay? Whatever size they are, they are. Some of them can potentially use some energy. They don't. There are certain times which you might say a virus moves. We're going to look at that, particularly during the infection stage of things. You might say they move. So I'm not going to. They definitely reproduce. They definitely do not respond to stimulus. So these two are kind of iffy. Like, This one definitely. They can't do any of this on their own. If you can't live on your own, you must have a host. What do you call it? What's that called? Whether it's a virus or a bacteria or a baby or anything.
Parasite?
A parasite. Did you catch I call a baby a parasite? All right. At any rate, if you teenagers, if you must have somebody else for your existence, that's a parasite, right? Viruses, they're a very particular type of parasite. Okay? They live inside of cells. They must be inside of cells. So we refer to them as intracellular. Intracellular. And they must have a host. No host, no viruses. So they're obligated. So intracellular, obligate parasites. That's a real good definition of these things. You make me nervous without any notes. There will be a reckoning, you know what I mean? It's going to come. You got to know all this. You very much likely need to put on a test, hey, either what does it mean when we say the virus is an intracellular obligate parasite? You tell me what each of those parts are. Viruses, viroids, prions, they're all similar, not the same. What do I want you to know about each one just a little bit before we go deeper? Let's look at their structure, okay? If we're going to compare these three, we got to begin with their structure, all right? We start with some, let's start with some stuff, okay? So, viroids equal nucleic acid only. Prions equal protein only. Viruses, nucleic acid and protein. It is crazy wicked hot here, isn't it? Don't mind opening that door. You might have to move to like prop it open or something. So viroids, nucleic acid only, prions, protein only, viruses, nucleic acid and protein. Let's walk through these. You may have just had all this last semester and you're like, I know exactly what that is. You may go, I don't know what any of these are. I haven't never had biology. That's okay. So nucleic acid. When you say DNA or RNA, the NA is nucleic acid. Deoxyribonucleic acid, ribonucleic acid. These are just the genetic code of a cell. Okay? It's called nucleic acid because we find this typically in the nucleus. Not that it's exclusively in the nucleus, but it's typically in the nucleus. And so what's weird about a viroid is they don't have a cell, they don't have a nucleus at all, but they'll have either DNA or RNA. Your book doesn't have much to say about these, quite frankly. It's got a little tiny bit on viroids. Let me just read this for you. Viroids are plant pathogens, so that's a thing to know. These guys really only infect plants. Small single-stranded circular RNA particles that are much simpler than a virus. So the nucleic acid here is RNA. And this RNA exists in a circle. Okay. So that's it. That's all this thing consists of. But when it infects a plant, the plant's protein ribosomes, they see this, and they go, oh, okay, that's RNA. I know what to do with RNA. I read RNA and I make proteins. They also have, it can reverse engineer itself, and it can end up making DNA and then making more RNA and reproducing. Well, between hijacking that plant cell's cellular mechanisms and the proteins that it produces, it creates an infection in that plant cell. They do not have a capsid or outer envelope, those are proteins, but like viruses can reproduce only within a host cell. Viroids do not, however, manufacture any proteins, They only produce a single specific RNA molecule. Human diseases caused by viroids have yet to be identified, so we don't know of any here. Viroids are known to infect plants and are responsible for crop failures and loss of millions of dollars agriculturally. That's probably the only reason we're talking about this at all. It doesn't affect us, but it does infect our food. And, so if you're having a garden and it got infected with the right viroid, you could lose a whole crop. Well, if this is what you do for a living, or what you want to do for a living, right, Thompson? This would be devastating. Okay? But all this is a simple little single strand of RNA. And it can spread throughout the plant, and it can spread from plant to plant to plant if they get infected with it. And it's like nothing. And yet it can cause all this damage. Another thing here is a prion. A prion is only a protein, and this is like zombie stuff. Thank you for that. It's scary. You ever heard of mad cow disease? Okay.
Yeah.
Yeah, or there's a disease, it's almost eradicated, called kuru. I believe it's from Papua New Guinea. So, mad cow disease, you get a cow, and it gets this little prion, this little protein in its brain, and that protein is slightly misshaped. What's wild is it's basically its own protein. It's a naturally occurring cow brain protein that becomes misshaped. And when it does, it's like a big old game of tag. As soon as it comes into contact with another of the same kind of protein, it changes that protein's shape. And now I've got two of them, and when they come in contact with others, well, now I've got 4, and then I've got, what's 4 times 4? Or 4 times 2 is 16, and then 32. So I'm growing exponentially, playing this game of tag, just converting natural, healthy cow brain proteins and causing them to change shape. And what this does, as these are all changing shape, is it changes, you get these lesions. All of a sudden the cow's brain becomes like it looks like a sponge. It's almost got these little holes and lesions running all through it. You need your brain, right? This is where you do your thinking, you're memorizing, you organize your activities, for how you function. So a cow gets infected with this, its brain's turning into a sponge, and it's going to die, right? Now, Farmer John has a dead cow, and I don't know, if in any way that brain matter from that first cow comes into contact with another cow, this might sound gross, but it's not that uncommon. What if you took your cow and you ground it up and you fed some to your cows? You didn't know any better. Well, you just infected your whole herd with this prion. All it takes is one of these little brain proteins that is converted. I just put it into my whole herd, and now in each one of them, once it gets to the brain, it begins converting all the others. And now all my cows have these spongy brains. That's scary. So I got this whole herd of cows that are just dying. What do I do with that? I make hamburger meat and I sell it. Right? Because they're not making milk anymore, they're not eating grass. Okay? And now I've just given it to people. It was a cow disease. Well, it turns out that when people eat cows that were infected with mad cow disease, they develop what's called Jakob-Creutzfeldt disease. And they have the same thing happen in their brains. And the brains turn to sponge and they die. And heaven forbid that you came into contact with that, the thing can spread. And all it is it's a naturally occurring protein that just got misshaped. It plays a game of tag, and you laughed at the word zombie, but it creates zombie cow kind of a thing, right? And they can spread it, and zombie people kind of a thing. And they didn't expect this to happen. We went through the coronavirus thing, right? Where do they think that started? Bats, right? So you would expect that a disease of bats would infect bats, but you don't necessarily think that a disease for bats would infect humans. But sometimes a disease can mutate, its DNA can change such that it can jump from host to host, a type of host to another type of host. So your cells are very different than bats, but they're not completely different. You have a lot of structures on your cells that are similar enough to a bat's cells that it's reasonable to think that maybe just the right virus could latch onto your cells and infect them. Not the most common, but this wasn't a virus. This is a prion. It's just a protein. You don't have cow proteins. You have human proteins. There's enough similarity between your human brain proteins and their cow brain proteins that it was able to infect them. And you know, because of this, like, people stopped importing beef from Great Britain where this was happening, and you know, it's scary stuff. So, viroids, nucleic acid only infects plants. We don't see, has a mechanism infecting plants. It doesn't seem to have a mechanism for infecting human beings. Prions, they seem to infect mammals, but we don't seem to see them in plants. You're not going to see this in the bacteria. There's a level of specificity about what kind of host these parasites can infect. These are on the rarer side of things. Viruses are everywhere. Everywhere. They're all over the place. The only question is, can you get them or not? Do you have immunity to them or not? Can you develop immunity to them or not? Okay? So we're going to spend the rest of our time talking about viruses. All right? All right. I'm going to erase some stuff and we're going to rock and roll. Which one has RNA only? Viroids. Which one has protein only?
Prion.
Prions. Good. Think P's, right? Proteins, prions. It's helpful. Okay. Well, let's go. Viruses. How have we described them so far? 3 words. Intracellular. Parasite's the last word. Obligate. They're obligated. They can't exist on their own. They can't replicate on their own, right? Alive or dead? Or never alive? Never alive. Not living. Viroid. Live, dead, never alive. Never alive. Prion, same thing, right? These things, they don't do all the characteristics of life. All right. Oh. I have hit the stage in my life I need reading glasses, or like little cheater glasses, but I've only just gotten there. So I have like 5 pairs at church for when I'm reading on the pulpit, but I have none here, and I'm sitting here going, oh man, this is a mistake. I gotta get to the dollar store. Okay, viruses are non-cellular. They don't have cells, okay? You ready with it? So the first thing we're gonna look at here is structure. First thing we want to know about a structure is they're non-cellular. They do have proteins like cells do. They do have nucleic acids like cells do, right? I'm going to draw 2 types, okay? There's going to be good examples for us. That's pretty good. This is called a T4 bacteriophage. You will not ever be tested to know that that's a T4 bacteriophage, but it is a T4 bacteriophage. It looks like this. This shape is called an icosahedron. It's made up of 20 perfectly symmetrical triangles. It's kind of cool. It has this. It's got a collar, or a plate. It's got a collar. It's got a base plate, and then it's got these little feet. This is its structure. Everything you see here right now is made out of protein, okay? All of this is protein. If you've not drawn this, please draw it. Okay? But I told you that these things are made up of proteins and nucleic acids. Where's my nucleic acid? Just if you're to take a guess, where's it likely to be?
The circle?
Not bad. Certainly going to have to get to the circle probably, right? But it's actually stored in here. Okay? I'm going to draw some nucleic acid in, I'm going to do pink. Why not? Okay. So, I've got this protein thing, I've got this nucleic acid thing. This protein thing has a name. It's called a capsid. Protein capsid. You can think of this as the body. You can think of this as the shell. We could draw an analogy to like a needle or like a syringe, like you would inject medicine through. Right? So this would be like the syringe, and then the nucleic acid on the inside would be, whatever it is that you're actually injecting. This is the outer part. This is the structural part. Every type of bacteria or of a virus has a protein capsid. Every single type of a virus has some nucleic acid. The nucleic acid is always stored somewhere inside of that protein capsid, but not all viruses look like this. This is actually probably the most complicated looking virus there is. Aren't you glad I made you draw that? I could have picked something easy, but I didn't. I don't know. This is just kind of classic. So what's really fascinating about these, and one of the reasons they're worth drawing, is if this thing were to come up on a cell, a bacterial cell, these little feet here, they find the right landing pad, so to speak. They have, everything in biology is about charge and shape. And so if this is positively charged and there's a positively charged region on that cell, that's not going to work because opposites attract. It needs to find a negatively charged space. If this is hydrophilic, in other words, water-loving, it's not going to match with a hydrophobic part. Conversely, if this is hydrophobic, it can't match with a hydrophilic part, right? Because opposites attract and like repels, and so this has to find the right landing spot. But once it does, it sets up, and then it acts just like a hypodermic needle, and it injects the nucleic acid into the cell. Okay. So the function of a protein capsid is all about nucleic acid delivery. How are we going to get this inside of the cell? Okay, that's its function. Let's say something about that. Its goal, its function is to get nucleic acid. Grandmother comes and knocks on the door. What are you going to do? You're going to open the door? There's some guy dressed in a mask who looks like he's going to rob you, comes and bangs on the door. What are you going to do?
Pretend nobody's home.
Pretend nobody's home, lock the door, call the cops, something, right? You're not letting that guy in. Well, here's the whole thing. This is a wolf in sheep's clothing. These things are viruses. They exist by being wolves in sheep's clothing. They look, at least down here where it's touching, they look like, oh, this is grandma. But it's not. It's the wolf. And so it's able to it's able to attach, latch, inject, take over, and destroy. Okay. All right. So they all have a protein capsid. They all have a nucleic acid, but some of them have more than that. Some of them have what we call an envelope. Okay. Give us a good picture for you. Goodness, is it hard? Yeah, here's it. Okay, let's do this. I'm going to give you another drawing. Okay. So, what color did I use for the human cell? Orange. And what's the outermost part of a human cell? Cell.
Membrane?
Membrane. Okay. Phospholipid bilayer membrane. Okay. So, crazy thing with like HIV or others, they have an envelope. So I'm going to draw that. What color am I drawing? Why? It's made, the envelope for HIV is made out of human membrane. Remember I told you these things are wolves in sheep's clothing. They want to get your cells to let them in. What better way to do that than disguise yourself as a human cell? And so something like HIV is going to have protein capsid, going to have nucleic acid, but it also has what we call an envelope. Okay? The envelope is the host membrane. We'll look at how they get this envelope later on, but the fact that they have one is a very, very big deal. Two things every virus has: protein and nucleic acid. And that protein forms what we call the capsid. The capsid is basically the shell, the structure, the mechanism of getting that nucleic acid into the cell. Guys with an envelope have a whole other level of sophistication in terms of deceiving cells and finding ways to get in. Okay? All right, so we've got this. We've got our protein capsid. And we've got our nucleic acid. I'm going to draw three things. This is kind of just a conceptual, where are we at right now, check. I want you to tell me what's what, okay? So I'm going to draw four things. All right, you got A, B, C, and D. What is D?
It's a virus with an envelope.
It's a virus with an envelope. What's A? Yes. Next level, correct answer is Viroid, right? Viroid has only the nucleic acid. Good. What's this? Prion. What's this?
Virus.
Regular run-of-the-mill virus. Okay. Every one of these is an obligate intracellular parasite. They can't replicate on their own. They can't respond to stimulus. They can reproduce with your help or some host cell's help. Some of them can kind of move, like this doesn't move, doesn't use energy. This doesn't move, this doesn't use energy. This definitely does. Definitely does. But, all right. So far, so good? Okay. Why are we talking about these? I don't know that I explicitly told you necessarily, but why do you think so far we're talking about this? Some reasons. Why do these matter for you? Because I've got so much content to cover this semester, I don't have time to give you stuff that doesn't matter. I can wear you out with stuff that impacts your life. So how does this impact your life? Yeah, this will get you sick. This will destroy your crops. This will eat up your brain and you'll be going like a crazy cow. This is HIV, right? I mean, not like that, but I mean, this is what HIV is like. This is what the common cold is like. This will destroy plants, but it's not a threat to you, like as far as your body. And none of these are alive, but they're kind of on the edge, right? I mean, well, it's got a lot of things that look like life, a lot of structures. None of these have a cell. Although that looks like a cell, doesn't it? Kind of. But none of these have a cell. These are also all, compared to cells, they're tiny. These are teeny tiny little things, okay? All right, well, let's move on. Let's look a little more at structure, okay? In particular, we're going to look at capsids. I'm going to inevitably throw some words at you that you may or may not know. I'm not going to apologize for that. I'm going to help you learn them. There's great meaning in any of the words that we cover, and that'll help you across all of life. Do you all know the word metamorphosis? What does it mean?
Like changing shape.
Changing shape. Which word means shape in that? Which part means shape in metamorphosis? Is it the meta or is it the morph?
Morph.
Morph. Okay. Morph refers to shape. So when I say morphological or morphology, we're talking about its shape in some way, like looking at, comparing, studying its shape. So we're going to look at capsid morphology, all right? All right. So we got four basic types. You've got helical, icosahedral, enveloped, and head to tail. All right. So let's draw these. When I say it is helical, I mean that those proteins that it's made out of do this. It's probably not the best drawing of this. It's not as good as I want it to be. But it forms a helix. A helix is just saying that it's doing this. Kind of like a spring or a coil. In fact, that's why didn't I just do that? I don't know. We'll do that. Okay? So this is helical. Now it's saying that the protein is helical. This is very, very common. This is not a virus yet, right? What's missing? Nucleic acid. And where will I find the nucleic acid? Inside. Okay. Now I got a virus. All right. Next shape is icosahedral. It's this kind of shape. Remember, it's 20 triangles put together. Not a virus yet. I'm missing nucleic acid. Okay. All right, the next type is enveloped. And the last one is head to tail. Most viruses are going to fit one of these 4 patterns. And it would be nice and it would be easy if we said, okay, helical, those will only infect mammals. Icosahedral, those will only infect fish. Envelope, those will only infect bacteria, but that's not true. Any one of these shapes could potentially be found as a pathogen of any of those other organisms. It's not so much the structure of the capsid, the way it's laid out, as how it attaches. It's how these little parts interact. Or does it have, so what mechanism does it have for getting inside of the cell? Okay? What's that affinity? And any one of these shapes can be successful in infecting. Okay? And one thing we haven't talked about yet is RNA versus DNA. There are types of viruses that infect humans that have DNA as their nucleic acid, and there are types that have RNA as their nucleic acid. Both can be successful. We see certain successful patterns in viruses, but it's not like it's just following down one exact path. In organisms that are alive, we're going to see patterns that are just so clear, like this is the way, this is the mind of God in creating these. We don't see that here. Okay? It's not like there's just this one pattern. Y'all feel good so far? Okay, good. Well, let's move on. Let's talk about viral receptors. I'm going to erase this side. If we were to look at a cell, oh, it's a lot of squiggles. What I'm drawing right now is referred to as the phospholipid bilayer. Not going to try to go back and explain all of that. If you do not know what I'm talking about, you might want to go look at it though. Okay. It's made up primarily of phospholipids. However, there are other things embedded in this. There are proteins, there are sugars, there are combinations of proteins and sugars. We might have like a gated protein, a channel. We might have a protein that's embedded, then has a sugar that's sticking off of it. These are fairly common. These are actually super, super important. Looks like I'm drawing quarter notes. But anyway, something like this would be referred to as a glycoprotein. These are incredibly, incredibly important. These are super, super specific. This is how cells recognize themselves. It's how they recognize foreign cells. If you get an infection, you get a bacterial infection, you want your white blood cells to roll into town, recognize this is not ours, or this cell has been infected with something foreign, we need to destroy it. Well, it does that by recognizing glycoproteins. If your cell comes up on something and you're like, that looks delicious, that looks like food, let's engulf that and bring it in. It's going to do that through receptors called glycoproteins. It works both ways. Because along comes a virus. And it's got just the right glycoprotein of its own. And it's able to attach, and it triggers, they don't all function the same way, okay, but I'm giving you an example. It's able to attach. Maybe there's a little negative charge right here, and a little positive charge right here, and there's a little positive charge right here, and a little negative charge right here, and those opposite charges attract, and this thing attaches. And now it creates what we call a signal cascade. And so this cell goes, hey, this is something we want. Let's bring it in. And it just brought in the wolf in sheep's clothing, thinking that it was doing something good. And so because of the charge and shape of this protein capsid, this virus just got in. Now, if this doesn't align just right with this particular cell type, host, organism, this isn't going to work. That's why typically you're not going to get bat viruses, right? But if that virus has a glycoprotein that matches with bats and also matches with humans, well, it's possible for you to get it. It's not only tends to be specific from like organism to organism, but also from cell type to cell type. You guys have heard of like cold sores, right? Anybody get cold sores? I'm the only person in this room who ever gets cold sores? Or canker sores, like the little sores in your mouth, okay? Where do you get canker sores? In your mouth. You don't get them on your wrist. You don't get them in your colon. You don't get them on your liver. You don't get them on your eyeball. Super, super specific to the cells in your mouth. HIV doesn't infect your liver, doesn't infect your brain, only infects your white blood cells. Super, super specific. Okay? Does that make sense? This is really, really important stuff. So this is a glycoprotein. This is often how viruses are able to attach and get in. Is they match up. They look like something that a cell might want to have, or else they just happen to be opportunistic, and here it is, and here it is, and like in this case, like the bacteria does not take this whole thing into itself. This thing opportunistically matches up its glycoproteins on its little receptor feet, and then just injects itself into its nucleic acid into that cell. And this stays out. It's really cool. The use of microscopes has improved over time. In the last century, we have pictures of this where a bacteriophage, by the way, that word means bacteria eater. Phage is Greek for eating. So a bacteriophage, when it infects it, this part stays out, and it just injects its nucleic acid. Which tells you really about how all of these work. It's the nucleic acid that's doing something. It's not the protein capsid. Its sole function is to contain it and to deliver it. All right. And this glycoprotein interacts with what we're calling a viral receptor. It's not like your cell wants to have a viral receptor. It's not like, I really need one of these. I'm feeling good, I'm feeling tough, I want to take on some viruses today, let me make this, put it on my cell, see if I can get somebody to come take on a challenge and fight it. It doesn't work like that. Your cell uses this for something entirely different, but the virus has adapted so that it can come and exploit an opportunity. All right. Do we have time?
10, 20.
20. How long do I have? I got 30 minutes?
30 minutes.
Yeah, okay. I probably won't finish, but I'm going to try. All right. Name some viruses.
COVID.
COVID, common flu. What else? HIV. What else?
Strep.
No, that's bacteria. We'll get to that. Those kind of things will be next. Any other viruses? Yeah, HIV, AIDS, chickenpox, herpes. Super specific. They impact specific cell types, right? Some of them you get, and you get it like once, right? If you got the flu last year, you're not going to get last year's flu again this year. You might get the flu again, but you're not going to get last year's flu. Typically, when you get chickenpox, how many times do you get chickenpox? Once. Anybody had chickenpox twice? I have.
My brother did.
Hmm?
My brother did.
You know anybody who got shingles? No? You ever heard of shingles? I understand it to be just a devil of a thing to get. Like, it just hurts. It's chickenpox. For people who already had it, like, it just comes back when you're older and you get it a second time. It's so much worse. But typically you get chickenpox once, and your body just destroys it, and you're immune to it. And we'll talk about that coming up. And typically you get the flu once, and your body learns to destroy it, and you move on. But other things you get over and over again. You said you get canker sores, right? You only had them once in your life, or you had them multiple times? Multiple, right? You get canker sores. You tell me if it's the same for you as it is for me. I am not going to get canker sores and everything's going great in my life. Everything's happy, everything's going my way. I'm not at all at risk for canker sores. If I get extremely stressed out during a very hard period of my life, I am really likely to get canker sores. Is that true for you? No rhyme or reason that you know of yet? Okay. Well, my experience is pretty typical for canker sores, because there are certain viruses that can go on and exist inside of your cell. You don't get immunity to them, but they go dormant until there's some kind of a trigger in the environment that signals for them to just boom, take off again. And it's fairly common for something like canker sores or cold sores that stress is a common stressor that will cause it to release. Man, it's hot here. Whew. Roasted. All right. Okay. It's a good place for us to be. We want to talk about infection. Okay. So ultimately, if a virus has no way of getting inside of you, or a viroid or prion, you don't care. Okay. It's only if it can infect you, right? So infection is its capacity to, or the activity of it getting inside of you. We want to look at infection, and we want to look at what happens after you get infected. What does that do to you? Okay. So I could draw a million pictures for you and make you draw a million pictures, but we'll never finish, and that's going to get very tedious. So I'm going to show you an animation, okay, that walks through this. I'm just kidding. All right. Let's do a split screen here. Okay, so I told you earlier that everything I show you will appear on Moodle, right? This is you guys. Hopefully you've already been here, done the syllabus quiz, stuff like that. So I've got you all right? Under topic 1, I've got 2 things going on. I have a lytic versus lysogenic cycle animation, and I have an immune response animation. We're going to look at what's called the lytic versus lysogenic cycle. Let's start with this. Your idea is infection. Once it's infected, it's going to go into one of 2 cycles, lytic or lysogenic. They're going to say lytic, that's fine. I don't know. It's probably more accurate to say lytic, but I like to say lytic better. Lytic versus lysogenic cycle. To lysis means to split or to break. The whole deal with a lysogenic cycle is it's probably going to kill that cell. It's going to go through, it's going to replicate in mass, and when it's time to break free, it's going to break free, and that cell is going to die imminently. Okay? Lysogenic. Lyso means to split, but genic means life. So before we split that cell is going to live. In the lysogenic cycle, it infects the cell, but it doesn't replicate right away. Instead, what it typically does is it incorporates itself into the host DNA. It hides out. That's what happens with your canker sores. It's in all those cells in your mouth, just hanging out, doing nothing. Trigger comes, and then it goes into the it leaves the lysogenic cycle and goes into the lytic cycle. Okay? So this is the big picture of infection. We get infected, and either we replicate and destroy right away, or we hang out for a while, and then we replicate and destroy. Okay? Well, here we go.
So let's talk about viral replication. Since we know that viruses are made of only proteins and one type of nucleic acid, which means they have no organelles to make copies of themselves with, they have to get inside other cells in order to use their ATP and their organelles to make copies of themselves. So first step, get inside that cell. And I'm just going to mention a few things for review. So remember, there are 3 ways of getting into a cell. One of the ways of getting in is that bacteriophages, they literally just inject their genetic material inside of the cell, whereas every other type of virus uses one of 2 ways. If they're non-enveloped, they trick the cell's receptors into letting them in, or if they're enveloped, they can either use that receptor trick or they directly fuse with the membrane. And once that genetic material is inside of the cell, the virus has hacked in and it's ready to do a takeover. But here's where it makes a choice. It's kind of either impatient and ready to get going to copy itself, or it sits back and says, you know what? I'd rather sit and wait until I'm ready to take over. So let's just draw these 2 possibilities out to see what happens. And with the first choice, the impatient virus goes ahead and takes over the cell's machinery, their ATP, ribosomes, nucleic acids, amino acids, to start making copies of the virus's genetic material, which again can be RNA or DNA, and also the virus's proteins to make their protein coat. And these will self-assemble. That means they just come together on their own to form fully functional viruses. And because it keeps making more and more and more of these, it will eventually force the cell to lyse or break open. And once it breaks open, all of these viruses are released into the environment. And if there are other cells nearby, then this army can start marching out to infect, to hack into those nearby cells and create more armies. So this makes a lot of sense if you have a lot of hosts around and your goal is to just create the biggest army in the fastest way possible. And so that's the impatient virus. So what about the other option? The other option is where the virus decides it's just going to sneak in and hitch a ride. It thinks that the bacteria seems to be doing fine on its own. Maybe there aren't other hosts nearby, so there's no reason to kill off the host because lysing it would kill the host, so we don't want that. We're just going to keep the host alive. And in order to sneak in and let the bacteria do its thing while it's waiting, it's going to combine with the host's genetic information so that the host really can't tell that it's there. It's basically quietly sitting there because it's repressed. There are repressor genes on this virus transcribed, so it's not doing anything. And this is called a provirus, or you might also hear the word prophage. And again, because it's not doing anything, this is called a dormant or latent phase. So the bacteria just does its own thing, it continues replicating, and of course the virus is already there, so it will continue to replicate. So it will replicate when the bacteria does, because it's part of its genome. But about 1 in 10,000 times that this happens, or if something happens like the bacteria is exposed to UV light or something, well, in any case, there's something that weakens that repressor gene that we talked about on that virus. So it's no longer quiet. And the host's genome, like most other genomes, will start wanting to repair itself. That means it will cut out to try to repair its genome. It will excise out part of its DNA that just happens to be the virus. And now the virus is active. It's ready to make copies of itself, lyse the cell, and get its army out into infecting other cells. So the official terms for the inpatient method is the lytic cycle, and the hitch-a-ride method is called the lysogenic cycle. So now you should have a good idea of what the lytic and lysogenic cycles are in viral replication.
I don't know if this came across well from that or not, but
Viruses are interesting because they are the
okay. In the lysogenic cycle, when one cell becomes infected, is that cell goes through the cell cycle, and it divides, and one cell becomes 2, and 2 becomes 4, and 4 becomes 8, and so on and so on. It's copying its own DNA each time to make a copy for each new cell. Well, if it has if it has the viral DNA that's incorporated in it, it's copying that along with it. So now 2 cells are infected, and 4 cells are infected, and 8 cells are infected, but the virus didn't have to do anything. The host cell is doing it for it. And again, that goes on until there's some kind of a trigger, and then it goes into the lytic cycle. Right? And again, this is why certain diseases can be dormant for quite a while. People who have herpes, they don't have a herpes outbreak all the time. Most of the time they don't. Most of the time it's in the lysogenic cycle until it goes through some kind of a trigger, and it'll go ahead and replicate and burst the cells. Other types of viruses, you get sick with it once, and your body is able to deal with it. While you're writing that, let me just tell you something. I'm not saying that all viruses have a mind or whatever. You know, obviously this doesn't. It's not alive at all. But a successful parasite does not kill its host. If you kill the host, you die too, typically. Viruses are a little bit of an exception because they can get out, but there are plenty. Orchids. You guys are familiar with what an orchid is? Real beautiful flowers, kind of exotic flowers. Vanilla. Vanilla beans, they come from the seed pods of a vanilla plant, which is an orchid. They can't live on their own. They're epiphytic parasites. They must live on, they're obligate epiphytic parasites. They must live on a host plant and draw nutrients and other things from that host plant. But if it's obligated, if it must have it, and then it kills the host plant, it kills itself. So Does that have anything to do with viruses? No, not necessarily, but that just is an interesting thing to think about for the idea of a parasite. A successful, a good parasite is not going to kill its host. So the free ride's over. All right, lytic and lysogenic cycle. What's the big difference?
One goes slower and one goes faster.
Yeah, one's right away, one does not. What's happening with the one that's not going right away?
Alright, you need the triggers.
It's hiding its DNA, okay? It's hiding its DNA, which is a successful strategy to go dormant for a while. There are a number of reasons that may be a successful strategy. But then eventually something's going to trigger it. It's going to reproduce. Now it's being reproduced all along anyways. As that cell reproduces, it's getting reproduced slowly. Then something triggers it and there's an outbreak, okay? And it goes crazy. These things can make thousands and thousands of themselves inside of a host cell. And then they burst, and now, one infected cell can turn into potentially thousands of infected cells. And what did it cost them? They're not using their own energy. They're not using their own natural resources. Their proteins come from the food that you ate. Their nucleic acid comes from your cells reproducing it. They don't have ribosomes to turn RNA into DNA, or excuse me, turn RNA into proteins. They don't have DNA helicase and polymerase and all these different cellular mechanisms for reproducing RNA. They don't have that. They just get you to do it. Highly, highly successful. And yet, they're not even alive. Okay? We tend to think simple or that complicated is better, right? I mean, everybody wants the latest iWatch and everything else, right? But if the power goes out, do you know what's going to work? A typewriter. It's not going to fail. You sit there and ding, ding. I mean, you can have a 200-year-old typewriter and it can still be going. You can get a 200-year-old, like, push the pedal sewing machine and you can still make your clothes. Right? But your watch has got to be charged. And heaven forbid you get it wet, right? Well, that's kind of an analog for organisms. Really complicated is cool. I mean, a virus is not going to do calculus, and neither am I, but, it has no capacity for that. But these things can survive what you and I cannot, right? A bacteria, and these aren't even alive, but a bacteria, super small, super simple, and yet they're everywhere. Great strategy. Simple is a very, very good strategy. Okay. So, I think we need to talk about an immune response. Because ideally, you don't die from a virus, right? Not that it's not trying to kill you. I mean, you understand the common cold is trying to kill you. I mean, it doesn't have a mind, but functionally, it's trying to replicate. It's actively replicating as quick as it can. If it does what it does perfectly, the common cold, then every cell in your respiratory system is going to be destroyed. And then you die. But you haven't died from it yet, right? I mean, people do die from it, but you haven't. And you've all had a cold, right? You've all had the flu. So how is it that you're not dead? That comes down to your immune system. Okay? So I want to show you some stuff with that. And again, you'll find these on the Moodle.
Whether it's fighting the coronavirus or the common cold, the body's immune system has several mechanisms to combat viral infection. So, how does the immune system respond to viruses? Before we explore this question in more detail, let's take a look at viruses and their effects on the human body. A virus needs to invade a living cell to replicate. A viral infection is when a virus invades the body and can lead to a spectrum of symptoms. The virus particle, which is called the virion, consists of a viral genome that is made up of either DNA or RNA and is enclosed in an outer protein shell called a capsid. Some viruses have an envelope around the capsid that contains lipids and proteins, including glycoproteins and viral receptor proteins known as spikes. The virion uses surface proteins in the capsid or glycoproteins in the envelope to attach to the host cell. After entering the cell, the virion turns it into a virus factory. The virus utilizes the host's protein formation and replication functions, which it uses to assemble into new viruses. Multiple copies of the virus are released from the host cell and spread to nearby cells where they repeat the cycle. So what strategies does the immune system use to prevent or curb infection? The first response to a viral infection is the rapid onset innate response. There are several mechanisms involved in innate immunity: interferons, natural killer cells, and antigen-presenting cells. When a virus infects a cell, the cell produces and releases signaling proteins called interferons, which are a type of cytokine. Interferons act as a warning signal to neighboring cells. They act by binding to interferon receptors on nearby uninfected cells. In response, the cells start producing antiviral proteins, which prevent the virus from replicating in the cell in the event of infection. Interferons also play another important role in the immune response. They induce nearby cells to increase the expression of major histocompatibility class I molecules, or MHC class I, on their surface. MHC class I molecules have two important signaling functions, for which they display endogenous viral fragments, known as antigens. First, they present antigens to adaptive immune cells, which we'll discuss later. Second, MHC class I molecules present antigens to innate immune cells. Let's look at this in detail. Infected cells are removed by specific lymphocytes that patrol the body. These natural killer cells, or NK cells, have receptors that recognize the antigens on MHC class I molecules. The absence of MHC class I molecules plays an important role in the immune response of NK cells. Usually, cells express sufficient MHC class I on their surface for T cells to be able to recognize them if they become infected. However, viruses can prevent infected cells from synthesizing these molecules. As a result, there are too few MHC class I molecules on the cell surface, which causes the infected cell to evade capture by T cells. The patrolling NK cells recognize the absence of MHC class I and release toxic substances, such as granzymes and perforin, which kill the virus-infected cell. Infected cells can also be removed via macrophages. These large leukocytes ingest free viruses in a process called phagocytosis. First, the macrophage surrounds and engulfs the virus. The macrophage then breaks the virus down by mixing it with digestive enzymes stored in vesicles called lysosomes. The degraded virus material is then released from the cell as waste. Macrophages are antigen-presenting cells, which means they're able to capture, process, and present antigens from pathogens. Another type of antigen-presenting cell is dendritic cells, which bridge the innate and adaptive immune responses. If the innate immune response...
We're going to stop. That was a whole lot, right? Let's see if we can make a little sense of this. A cell gets infected. Okay. Once it gets infected, it's inside, it starts to produce parts. It's a little virus factory. Cell recognizes there's a problem. Activities are not happening the way they're supposed to, and so it releases little substances called interferons. These are little chemical signals. These are warning signs to neighboring cells, okay? So that neighboring cell has a receptor and it recognizes that interferon and it causes some changes to that cell. That cell is going to begin one of the things they talk about is going to start producing major histocompatibility complexes, MHCs. So what? This is a wild, wild thing, okay? So MHCs begin pointing in. And at least a lot of them do, maybe not all. But if it comes into contact with something that's not supposed to be there, like a viral particle. I guess I should have drawn these in blue. Anyway, then what happens is they pop out and they show it off. You ever seen how a cat or a cat might walk up to you and go, here, look at this bird I just killed. You know, they're very proud of it. That's kind of the idea here. It's showing off, hey, there is something inside of me that is not good. And it's a danger to everything else. I need to be killed for the good of all the other cells. And so now your immune system, your white blood cells would come along and they would destroy this cell. Okay? So this is part of what we're calling the innate immune system. These cells are signaling for your body to destroy them so we can stop this infection in its tracks. Okay? There are other steps to all this. What time do we have? Not time for what's left. All right. We will pick up with the body's immune response next time, which is good because we'll be transitioning from viruses, viroids, prions, a little bit of immune system stuff, to bacteria. Between viruses and bacteria, that covers a whole lot of the pathogens, the diseases that affect humans. And so it's a good point for us to look at, well, how does your body respond to these? Why is it that you're not just dying at every little infection that you get? All right? God bless y'all. Have a wonderful day. You okay? Gotcha.